We will continue with the formation of thyroid hormones. We have seen first, in the first step is iodine trapping. The second step is the synthesis and the secretion of thyroglobulin. And the third step, the oxidation of iodides. And the fourth step was the organification of thyroglobulins. And the enzyme used is TPO, thyroid peroxidase, also called as thyroperoxidase. And this enzyme iodinates the tyrosine residues of thyroglobulin. If it iodinates at the third position, it forms monoiodotyrosine, and when it iodinates the fifth position also, it forms the 3,5-diiodotyrosine. In the next step, coupling occurs. One monoiodotyrosine reacts with one diiodotyrosine to form triiodothyronine T3, the more active form. DIT, diiodotyrosine, can couple with DIT to form T4 tetraiodotyrosine, which we also called as thyroxine. We have to remember that these coupling reactions are, when these coupling reactions are occurring, it is still bound to thyroglobulin. It is still bound to thyroglobulin. See how it is bound to thyroglobulin. It is bound to thyroglobulin in this format. See, this is the structure. See, this is the tyrosine residue of thyroglobulin, and here triiodothyronine. This is DIT, this is MIT. So if DIT and MIT couple, then this is triiodothyronine. If both DIT and DIT couple, it forms T4 thyroxine. This is reverse T3, which is an inactive form. So tyrosine, the coupling reaction of MIT and DIT produces T3 and T4. In the step 6, storage. Now this is very important. Endocrine glands do not store their secretion inside the gland, but this gland, thyroid, is an exception, since the secretions of this gland are stored. This T3, T4, they are still bound to thyroglobulin, and they are stored in the colloid. And this iodinated thyroglobulin can meet the body requirements up to 1 to 3 months. So if a person is having a diet deficient in iodine, the thyroglobulin, which is stored in the follicles, can form the T3 and T4 and release them into the blood, since it is stored. So the storage of thyroglobulin is a very important phenomenon, and such type of storage is not found in other endocrine glands. So these are the steps of formation. We will just review this. The steps. First, the structure and function unit of thyroid gland is the follicle, and the follicle cells, they will synthesize T3 and T4. The follicle cell, these are the follicle cell. The first step is the iodine trapping. The iodine which is present into the blood is transported into the follicle cells by the help of sodium iodide sympathers. These iodide ions are then transported into the colloid by iodine chloride transporter, which is called as the pendrin. And here thyroid peroxidase will oxidize this iodide. Two molecules of iodide, the negative charge will be removed to form iodine. Similarly, the rough ER present in the follicular cell will secrete thyroglobulin into the colloid. So in the colloid, now we have thyroglobulin and iodide. So iodine trapping. Second is the synthesis and the secretion of thyroglobulin. We have seen that rough ER will secrete the thyroglobulin into the colloid, and this is a dimer glycoprotein, and it has approx 140 tyrosine amino acid residues. In the third step, the iodide is oxidized into iodine, and in the fourth step, the thyroglobulin having tyrosine residues, the iodine will uh, will be attached to the tyrosine residues. If one iodide is attached, then it is called as monoiodotyrosine, and if two are attached, iodination is done at 3 and 5 both position, then it is diiodotyrosine. In the step 5, coupling of MIT and DIT will occur to produce T3, and if two molecules of DIT couple, then it will produce T4, which is still bound to thyroglobulin, and this thyroglobulin is stored in the lumen of the follicle cells and can meet body requirement up to 1 to 3 months. So this is how the hormone is stored. In the next lecture, we will see how these are secreted back into the blood.